Good morning. This is a stormy Monday morning whenever you're looking at this, probably later in the week. But here's a few practice things. I hope you're doing well. This is the first couple are, again, multiple choices that are good for whatever practice if you just do what they ask you to do, like describe in this case. Okay. So, again, the way we're doing these is take a look, scratch down your answer, and then go to the next slide and I'll walk you through it. So here we go. Okay, so in this one, they described to you one of the ones that was an example, if you remember, of long distance communication between cells. And that was insulin, and that was about sugar levels, glucose levels. So here the story they tell you is you have a liver cell, it's got a receptor, right? Insulin binds to that. Make insulin look like that. Will bind to that, right? Where did insulin come from? It came from the pancreas. They don't say that in your story, but this is what makes it long distance, right? It's going from the pancreas to the liver through the bloodstream. And this receptor then gets activated. It stimulates a phosphorylation cascade. They expect you to remember that's part of a signal transduction pathway. They could have also said that. And that causes the translocation, moving somewhere else, of glucose transporters from in here to the plasma membrane. We told you this story before, and we told it in terms of also the ADH hormone and kidney cells, which it causes aquaporins to get transported to the membrane, right? So there's the response. The response is that. And then when these have sugar transporters on the membrane, the sugar can come in and your blood sugar gets lower, and that's what insulin does, right? Which of the following then best describes the role of insulin? Very simply, it's the signal molecule, right? Which is what all hormones are. It's the signal molecule, because they never told you that this thing signals this to happen. They just told you that it sticks to this. And simply what they want you to say is, oh, the thing that sticks to that, do you remember what they called those things in general? Also, ligands, right? The thing that sticks to that, that is the signal molecule. So, ready for the next one. Okay? So now this is again multiple choice, right? I put the answers here just so I could point to. So they tell you a story here about these platelets, which are fragments of cells. They're normally around in the blood, right? They don't stick to blood vessel walls, but if they're damaged, right, these protein molecules in the wall are exposed to the inside of the blood vessel, and the exposed fibers and chemicals, right, release chemicals, these little dots, attract platelets. That must be, again, some kind of signaling kind of thing. They're telling a cell communication story here, right? What's the result? These platelets come, they start to form a plug and release other chemicals. I didn't like the specificity of this question, right? But this looping little arrow here in the diagram also suggests that these platelets are releasing more of the chemicals that cause more platelets to come around to clot here. So something causing more of itself to happen, that's a positive feedback thing. Okay? Negative feedback would be that this happens and this results in less of that happening, and then less and more and less and more. This is a more platelets here. So the best description of this is positive feedback. And the reason this one's not right is because, yes, it does result from interactions among things, but that's not what defines it as positive feedback. It's this few attracts more, produces more, results in more. That's what makes it positive feedback. Okay, next one. So this is a story of a very common neurotransmitter. In fact, our next question is going to look very similar to this because it's also going to have acetylcholine as its subject, right? But what they ask you here is about a signal molecule, a neurotransmitter This is the kind that's over short distance, right? Because even though this presynaptic cell and that postsynaptic cell connected by this synapse, the direction is not connect. There's a little space in between that makes this, again, also called a local regulator, this neurotransmitter, since it's between nerve cells, right? It can activate, right? So this means, again, we're sending a signal, activate an action potential. So he's investigating a thing that causes it to increase. That means the signal would increase. This neurotoxin is going to cause these nerve cells to keep saying, go, go, go. All right. He proposes two models now. Here's where it gets a little confusing. For a chemical that has this signal molecule in its name, acetylcholinesterine, but this is an enzyme. This is not a signal molecule, and this enzyme breaks down our signal. So that's why it would be good to fight this neurotoxin, because the neurotoxin says keep doing your signal, but this enzyme will break down that signal, therefore nullifying the effect of the neurotoxin. So that's the story they wanted you to put together, right? And he's got two models here. And this then is all about what kind of signal molecule is this? Is it one that binds to a receptor on the plasma membrane, or is it one that binds to a receptor inside like the lipid ones? Well, this diagram and up here shows that this binds to a membrane protein. Therefore, if in model B, right, in model B, you put this acetylcholine breaker downer in the postsynaptic cell, it's not going to bump into acetylcholine because it's not in the cytoplasm of the postsynaptic cell. So model A is going to be effective, right? Model B not effective. Notice this. They asked you two command verbs, predict and provide reasoning, for each of these models. So they're really asking you four things, but there's only 2 points here. So not only did you have to say, yes, it's going to be effective, but you also had to link that effectiveness to the correct reasoning to get even 1 point. All right. So make sure, again, you focus on the command verbs, do each one of them. That's why they put them in bold print. Then that's why they made this big letters, because they want you to talk about both the models, not just one. Next one, which again is going to sound similar because it's acetylcholine related, but it's really a different question. Okay. So now what really would make this a little potentially confusing is piggybacking it with that last one, because now they're talking about the receptors and the fact that there's different kinds of receptors, which is not uncommon at all, right? So here you have the idea that this acetylcholine signal molecule can bind to both of these kind of receptors, can bind, right? Which means the part of the receptor that this binds to must be really close to the same shape in both of these. But not exactly, because there's another molecule that can bind to one of them. Why would that be? They didn't ask you this, but why they could. Why would that be? Nicotine must be pretty similar to acetylcholine in its shape, but not so similar that it can bind to this one like acetylcholine can. So that's kind of the story, and if you put that story together, the questions aren't too difficult. Notice here what they did. Describe again, so don't give me any reasons, just describe the difference in both structure and function, right? So you need to address both between type 1 and type 2, the difference between type 1 and type 2 in both structure and function. Okay, so I was on this question, and I was part of the group that made up this rubric, and I had a disagreement with this rubric, but I understood why it was made like the way it is. I just tried to get this point across with somebody who didn't see it the same way as I did. Here's my point. Structure and function are two different things. That's why they're two different words, right? When you're asked to describe the structure of something, you don't tell what it does. That's a functional thing. You don't tell what it does. Sorry. you tell what it's made of, what shape it is, does it have charges here and there. That's what structure's about. So what I hope you said on this one was that, ooh, it looks like the binding sites here must differ in shape, right? What's my functional difference that results from that, which really is like, what's the reason why I said the binding sites must be different in shape? Well, it's because nicotine can bind to this one, but not to that one, so they must have a different shape, right? So this is probably what you said, and you probably said it more specifically, that nicotine can bind differently to type 1 and type 2. It can bind to type 1, not type 2. I know this is getting a little messy. Okay. Notice this. You could have also said, even though they asked you what the difference was, what's the function of these things? The function of these things is to activate receptors, cause muscle contraction, depolarize. So you could have said, well, there's no difference in function. Both of these type 1 and type 2 things result in the same thing: muscle contraction, depolarization, open channels. So you could have said that. It's unlikely that you did, because this is like the number one answer that like they were hoping that you said, basically. Okay. What could you have also said? Here's how these work. We read a bunch of papers before we start to make this up. All right. And we see what students have answered. And a lot of them talked about a function when trying to talk about a structure. Enough of them to where we said, okay, we're still going to give them a point for that. But notice this. This exact same sentence is also in the function column up here. But you couldn't say this twice and get 2 points for saying the same thing for structure and function. And that's what this says. You can only earn points from one row. So if you said this as a structural comment, you had to say this as a functional difference. Notice that this functional difference is also up here, right? And notice that the other thing you could have said for a structural thing is also in this functional column somewhere, right? So, a messy rubric it was, kind of, but really a simple kind of thing. These two have different shapes, but they're not quite so different that they both can't bond to this, but they're different enough that they both can't bond to that. Part B. Okay, so in this one, it's back to this kind of table again. Now they talk about the enzyme that we talked about in that other question, not the receptor, but the enzyme. And they tell you it breaks down acetylcholine in the synapse, right? Describe the effect of inhibiting this on the muscle cells with ACR type 2. Okay, well, acetylcholine can stimulate those and therefore stimulate muscle contraction and depolarization, right? Here's where a lot of students miss this one. They saw the word inhibiting and they said, oh, the muscle cells must be inhibited. No, what's being inhibited is the enzyme that breaks down the thing that activates the muscle cells. So if you don't break down the activator, that means you continue activating. So there's your description. This would be like a more detailed description of the same thing, but that's all you had to say. That was just part B, was just 1 point. Okay, I hope that was I hope that made sense to you, and I hope this makes sense to you. I'll bet your room's not messy, because if you're like me, your house has been cleaned more times than it ever has before in the last 4 weeks. And so I hope you have had a wonderful week, and I'm going to get back to you with a possible little test for this little unit. So long.